Okay, so now we're going to move on to chapter 8 and talk about smooth muscle. So to remind you, there are three types of muscle tissues found in the human body: skeletal muscle, which you've learned about in length, and we just discussed neuromuscular transmission at the neuromuscular junction between a motor neuron and skeletal muscle cells. And then now we're about to talk about smooth muscle, and then there's also cardiac muscle, but you will get this in more detail in future chapters. So what are some differences between skeletal and smooth muscle? Skeletal muscle, are attached to the skeleton and allows for movement of the entire body throughout the environment, whereas you'll find smooth muscle mainly lining the walls of internal organs. Skeletal muscle is voluntary, meaning that you can have conscious control over, you can decide to contract certain muscles, right? Whereas smooth muscle is completely involuntary. You have no control over the contraction of the smooth muscle in your intestines or in your blood vessels. That's all involuntary. So that goes along with the next point made. And then the overall cell shape is very different between the two. So in skeletal muscle cells, the reason why they're called fibers is because they're actually, a single skeletal muscle fiber is made up of multiple cells that throughout development form a syncytium, and so they fuse together. That's why you find multiple nuclei per skeletal muscle fiber. Whereas in smooth muscle cells, they're a little shorter. They have that spindled shape where you have a larger middle portion with tapering ends, and each cell contains a single nucleus. Skeletal muscle, you have striations because of the arrangement of actin and myosin in those sarcomeres, which you learned about. Whereas in smooth muscle, we still have actin and myosin. You'll see that those are myofilaments that are required for muscle contraction, but they're not arranged in sarcomeres, and so you do not get that striated appearance, hence why it's called smooth muscle. Skeletal muscle, you cannot self-stimulate. These skeletal muscle cells cannot be self-stimulated. It absolutely requires motor neuron input, whereas smooth muscle cells can be self-stimulating. Skeletal muscles, I just said this, but under the regulation of the nervous system, like skeletal muscle contraction cannot happen without motor neuron input. Whereas in smooth muscle, we'll talk about the fact that you can have nervous input, you can have endocrine or hormones, you can have stretching, and various other chemical and tissue factors that can all cause smooth muscle contraction or inhibit smooth muscle contraction. So we get a lot of different highly variable inputs for smooth muscle cells compared to skeletal muscle. In skeletal muscle, there's a high energy, lots of ATP required, and that's because of the high speed of contraction. Cross-bridge cycling happens more frequently in skeletal muscle, therefore we have a higher energy demand, whereas in smooth muscle, we have low speed contraction, fewer cross-bridge cycles, lower energy requirements, which makes sense if you think about it. And in smooth muscle, we can have rhythmic contractions. You'll see this especially in like the gut and the intestines, where you have the nice peristaltic waves, rhythmic contractions. And I will hit on this last part in one of my last slides as far as the stress releasing response, stress relaxation. And then skeletal muscles, they easily fatigue, whereas smooth muscles do not easily fatigue and can maintain prolonged contractions for quite a long time. They're capable of that. So I as far as comparing and contrasting skeletal muscle and smooth muscle, I'll talk more about some of the important comparisons throughout the next couple of slides. I in no way am gonna, it might be a multiple choice question where I give you a characteristic, right, of a particular muscle type, and you have to tell me, is it smooth or skeletal, maybe something like that. So in general, there are two types of smooth muscle. There's multi-unit smooth muscle and unitary or single unit smooth muscle. So multi-unit smooth muscle, so we're going to focus on the left image right here. You'll notice that these smooth muscle cells, there are few, if any, electrical connections between the cells. So this cell is going to act independently of this cell and this cell and this cell, right? So their contractions are independently regulated, right? And so this allows for a greater control, and mainly the control is exerted through the nervous system for multi-unit smooth muscle. But because each smooth muscle cell contracts independently, you have a greater degree of control and finer control over this type of smooth muscle. And you find multi-unit smooth muscle in the ciliary body of the eye and the piloerector muscles of the skin. So these are the muscles that are attached to hair follicles and in some blood vessels. But mainly when we're thinking about smooth muscle, you most likely are thinking about unitary smooth muscle. And sometimes it's referred to as syncytial smooth muscle. You'll see that the reason for that is because you'll have hundreds or thousands of smooth muscle cells that act as a syncytium or a unit, and that's because they will be connected functionally through these gap junctions. So these specialized cell-cell junctions that are like little channels that allow ions to flow freely from one cell to another. So when we get an action potential in one area, it's going to travel very quickly throughout lots of these smooth muscle cells, and we get a coordinated contraction, and that's why sometimes it's referred to as syncytial smooth muscle. Or if you may have heard it referred to as visceral smooth muscle, and that's because you find this unitary smooth muscle in the walls of hollow organs like the GI tract, uterus, bladder, and many of the blood vessels. So I think I hit on most of the points, but definitely remember that unitary smooth muscle, you're going to have lots of smooth muscle cells that are acting in a coordinated fashion because they're connected, they're electrically coupled through gap junctions, okay? So the anatomy of smooth muscle, I didn't lecture, but I think you got a previous lecture that went over the sarcomere, so myofibrils, and that sarcomere structure that you find in skeletal muscle cells. Now in smooth muscle cells, we have actin and myosin, but it's not arranged in sarcomeres, which I mentioned on that very first slide, comparing and contrasting skeletal muscle and smooth muscle. So here we have actin filaments, which are the thin filaments, myosin, which are the thick, and instead of being arranged in a sarcomere fashion, you'll notice that the actin filaments are anchored to these protein structures called dense bodies, and these dense bodies are similar or analogous to Z discs. So it's what these actin filaments are anchored to. You will see cross-bridge cycling happen in smooth muscle cells. I will mention a few differences in the next slide. But you do see cross-bridge cycles. But another key point that I want to make on this slide, as far as something to be aware of and know, is that if you were to go back in your lecture and look at about skeletal muscle, like sarcomere structure, and you looked at the arrangement of the myosin heads, they would be oriented in the same direction, both above and below, right? Whereas here in smooth muscle, we see our myosin heads going in opposite directions. And this anatomical feature of the actin and myosin like structure it's this feature that allows for cells to contract as much as 80% of their length instead of being limited to less than 30% like in skeletal muscle. So it's this unique structure here that allows for a greater force of contraction in smooth muscle cells. And then another point to just mention again is that smooth muscle contraction, because it's low energy and we have fewer cross-bridge cycling happening, we can have very long, prolonged contraction. Again, what I talk about is what you will be responsible for. So the extra information on these slides is just there to help you if you want extra information. contraction of smooth muscle. All right, so how do we get smooth muscle to contract? So in smooth muscle cells, smooth muscle cells are going to rely heavily on extracellular calcium. We talked about in the skeletal muscle cells how they have extensive sarcoplasmic reticulum, which stores calcium. Well, in smooth muscle cells, there's not really like a lot of sarcoplasmic reticulum. There's some, and some smooth muscle cells might have a little bit more than others, but for the most part, it's not very extensive. But calcium is still required for smooth muscle contraction. And so the smooth muscle cells have to rely more heavily on extracellular fluid calcium. And so any imbalances in extracellular calcium levels can throw off smooth muscle contraction because they don't have extensive sarcoplasmic reticulum. All right, so some sort of stimulus arrives, right? Whether it's going to be nervous input, stretch, it can be hormonal or local tissue factors. Whatever the stimulus is that arrives at a smooth muscle cell, if we're going to get smooth muscle contraction, if it's not an inhibitory signal, right? If it's an excitatory signal, we're going to get something is going to cause these calcium channels to open in the plasma membrane. Or I'll mention in another slide, certain signals can act directly on these calcium channels in the plasma membrane that allows extracellular calcium to come in. We can also have some signals that will act on the calcium channels of the sarcoplasmic reticulum, and that's how we get calcium entry into the cytoplasm. Either way, so we have calcium come into the cytoplasm of the smooth muscle cell. And instead of calcium binding to troponin, like we saw in skeletal muscle, we don't have troponin here. Calcium will bind to a calcium binding protein called calcium calmodulin. Calcium calmodulin will then, once bound by calcium, will activate myosin light chain kinase. And a kinase is an enzyme that phosphorylates substrates. It phosphorylates things. And in the name, myosin light chain kinase, this is an enzyme that will phosphorylate the regulatory chain in myosin. So it's going to go and phosphorylate the regulatory chains of myosin, and once these myosin heads are phosphorylated, now we can get the cross-bridge cycling happening, and it can interact with actin, we can get cross-bridge cycling, and we can get muscle contraction. All right, so unlike skeletal muscle, we don't have troponin here, right? Calcium will bind calmodulin. Calmodulin activates myosin light chain kinase, which then phosphorylates myosin. And when it is phosphorylated, it can interact with actin. We can get cross-bridge cycling happening, and we can get muscle contraction. So how do we get relaxation of smooth muscle? So in skeletal muscle, we got relaxation by very quickly degrading acetylcholine away from the synapse, right? Acetylcholine diffusing away or being degraded by acetylcholine esterase. In smooth muscle, in order to get relaxation, we have to have another enzyme come into play called myosin phosphatase, okay? And what a phosphatase does, it's an enzyme that will cleave off phosphates from substrates. Right? So myosin phosphatase comes and it will cleave off the phosphates, and that is going to in turn, like myosin can no longer form cross bridges, right, with actin. And then at the same time, we have some calcium ATPases that are going to be active. You have ATPases in the sarcoplasmic reticulum, as well as calcium ATPases in the plasma membrane. And what these guys do, they use ATP, so they require energy, to pump calcium out or into the sarcoplasmic reticulum away from the cytoplasm so that calcium calmodulin can no longer exert its effects on myosin light chain kinase, right? So that with the pumping out of calcium and the activity of myosin phosphatase, we can get muscle relaxation. Okay? And so really how quickly smooth muscle cells can relax will be dependent upon the levels of myosin phosphatase present in the cells, as well as the activity of these ATPases. Now with smooth muscle, if we're talking about smooth muscle being innervated by the nervous system, the neuromuscular junctions look a little different from the neuromuscular junction we saw between a motor neuron and the skeletal muscle cell. Up here, this first image, this is single unit, or that also called unitary, syncytial, or visceral smooth muscle. Right, so we're just looking at single unit smooth muscle from the wall of the intestine. You'll notice that these smooth muscle cells are connected. They're electrically coupled through those gap junctions. So again, single unit or unitary smooth muscle. And instead, like with skeletal muscle, we saw a single synaptic knob terminating on a single skeletal muscle fiber, right? Whereas here, what we see is along the length of the axon of this particular neuron, you see these varicosities, and each one of these is kind of like analogous to a synaptic terminal or a synaptic knob of a motor neuron, right? And each one of these will have neurotransmitter vesicles present and capable of releasing neurotransmitter that can then diffuse across the extracellular fluid and go affect receptors on the smooth muscle cells. Okay, so it looks a little different, but the overall idea is still the same: is that neurotransmitter gets released by these varicosities. Okay, and I will just point out, so it says that autonomic neuron, smooth muscle is going to be innervated by the autonomic nervous system, not the somatic nervous system, right? Because it's involuntary contractions that are happening. And then below this image down here, this is a multi-unit smooth muscle, so like in the ciliary body of the eye or the piloerector muscles of the skin. Here we have our autonomic neurons. These axons are going to come down. Again, we see those varicosities, which will all contain neurotransmitter vesicles. And I'm just going to highlight that point again, that in multi-unit smooth muscle, they're not electrically coupled, but rather they are independent. Okay? The main neurotransmitters involved in smooth muscle contraction are going to be acetylcholine and norepinephrine from the autonomic nervous system. And so here I'm not going to ask you to memorize the structures of acetylcholine and norepinephrine, but the point I want to make here is that acetylcholine and norepinephrine can both be excitatory and inhibitory. It depends on the smooth muscle, and you might ask, like, how is that possible? So when we talked about skeletal muscle, right, we only talked about the only neurotransmitter involved in skeletal muscle contraction is acetylcholine, and acetylcholine is excitatory always. There are no inhibitory neurotransmitters with skeletal muscle contraction. In smooth muscle, that's different. We can have neurotransmitters, either acetylcholine or norepinephrine, and they can be excitatory or inhibitory. It depends on the receptors that are present, right? So if it's a receptor that causes inhibition, or if it's a receptor that causes excitation, we can get different results. So I hope that makes sense. It depends on the smooth muscle, where it's located, and what receptors are present, whether acetylcholine will have excitatory or inhibitory, or if norepinephrine has excitatory or inhibitory. Okay? So it all depends on the receptors that are present. All right, so with this slide, the main thing that I want to point out here is that when we so in the skeletal muscle contraction, remember the plasma membrane of skeletal muscle cells are capable of conducting action potentials, just like the axons of neurons are capable of conducting action potentials. And if you were to look at the profile of the action potential on the sarcolemma or on the axon of a neuron, the action potential profile would look like this, right? It's just a single spike potential, right? That's what it looks like on skeletal muscle cells. That's what it looks like on neurons. Some smooth muscle cells will also have an action potential profile very similar to skeletal muscle and neurons. But we can also see other types of action potential profiles. So that's a difference between our smooth muscle and skeletal muscle, is that we can have different types of action potentials present. Right, so we can have this spike potential. Again, it looks very similar to skeletal muscle or a neuron. We can also have smooth muscle cells smooth muscle that's capable of this action potential profile, where we have these slow waves, these kind of undulating depolarization, repolarization. And when we get to the top of this depolarization, that's when we can have action potentials actually sent. I only bring this up because when you talk about the intestines, and when you get to the GI system, this type of profile is what you'll see in intestinal smooth muscle. And it's these slow waves with these spike potentials on top that gives you that rhythmic contraction of the intestines. So I only bring that up now just because you'll see it again later. I don't think I'll ask a question about this. The third type of action potential profile that you could see in smooth muscle is an action potential with a plateau. All right, so let's talk a little bit more about this plateau. All right, so the depolarization phase, that rise in positivity, is going to be similar to what you see here. But the repolarization, rather than being very quick in repolarization, we have prolonged repolarization, and that is going to be due to special calcium channels called L-type calcium channels. And this is going to allow calcium in, but at the same time we have an outward flux of potassium, which kind of balances the membrane potential, and it prolongs the ability of the cell to repolarize, right? And what's the point? Like, why would we want to have this plateau phase where we have calcium coming in, but potassium leaving? We're not really depolarizing anymore, but we're not repolarizing, but calcium's still coming in. Well, what do smooth muscle cells rely on? Calcium coming in for contraction. So this plateau, when you see smooth muscle cells that exhibit this type of action potential profile, it allows for more calcium to come in and prolonged contraction. Okay, so that's the key point here. You will see an action potential profile similar to this when you talk about cardiac muscle, cardiomyocytes in future chapters. All right, so remember I talked about how in skeletal muscle, in order to get contraction of skeletal muscle, we have to have nervous input, and it's the neurotransmitter acetylcholine. Well, in smooth muscle, we can have nervous input, so we can have neurotransmitters that affect smooth muscle contraction. We can have local and chemical tissue factors. We can have hormones. We can have stretch. So this slide just talks a little bit about that. You can read this if you want. I'll hit more on this in just a second, but you can have stretch that causes smooth muscle contraction, and this is very prevalent in the smooth muscle of the gut, of the intestines. You can have hormones, say like adrenaline, you can antidiuretic hormone. So different hormones can affect smooth muscle contraction or inhibition. You can also have local tissue chemical factors. A good example of this is your blood vessels, like low oxygen tension, increased carbon dioxide, increased acidity, or an increase in body temperature. These are some of the key ones that I wanted you to be aware of. All of these factors, right, changes in oxygen especially, so lack of oxygen, will cause blood vessels to dilate, right? So they will relax, they will dilate so that you can get increased blood flow to that area. That makes sense, right? So like if you think about it, just for this first one, if there's a lack of oxygen, lower oxygen tension, we want more oxygen there. So we want to relax the blood vessels, increase the diameter so we have more blood flow and more delivery of oxygen to that area, right? So that's just an example I wanted to bring up as far as chemical factors that can affect smooth muscle contraction or relaxation. I stole this image from the internet, and I thought it was just another way to show you, so over here, this is on the left side, this is multi-unit smooth muscle. Right? They're not connected through gap junctions like you see over here, so they're all independent. We can have, here's an autonomic neuron with those varicosities, right? And so you can have neurotransmitter being released here, which can be like acetylcholine or norepinephrine, and depending on which receptors they bind to, could cause contraction or inhibition of contraction, right? Over here, same thing. This is our single unit or our visceral smooth muscle. They're connected by gap junctions, so they're electrically coupled. They act as a syncytium. Here's our autonomic neuron with this varicosity containing neurotransmitter vesicles. We can get neurotransmitter being released, and again, depending on what receptors that neurotransmitter binds to, we could have an increase in calcium and contraction, or we could have inhibition of contraction. Just depends on which receptors are being bound, right? Here in the middle, we see this as a capillary delivering blood, and so different local tissue factors can diffuse out of that blood and go and affect receptors on the smooth muscle, and we can get contraction or inhibition of contraction that way as well. So I just thought it was a good slide to show you. I in no way, shape, or form am going to ask you all the details of this. It's just again to highlight the point where in skeletal muscle, we have to have nervous input. We have to have acetylcholine causing contraction, and there's no inhibitory neurotransmitter with skeletal muscle. But in smooth muscle, we can have, I already talked kind of extensively about the nervous input that you can have with the neurotransmitters being released, right? We can also have stretch channels being activated. So you'll see this in like the myogenic response in blood vessels. You can have stretch causing the opening of calcium channels and the influx of calcium in. You can have neurotransmitters, okay, that affect smooth muscle. You can also have hormones or those local chemical tissue factors. And they can either open calcium channels directly on the plasma membrane, or they can operate through second messenger systems to affect calcium channels present on that sarcoplasmic reticulum to release calcium. But either way, if we're going to get smooth muscle contraction, we have to have an intracellular rise in calcium concentration so that we can have calcium calmodulin activate myosin light chain kinase to phosphorylate myosin, and then we can get our cross-bridge cycling happening, okay? So really, I wanted to highlight how different the smooth muscle, how we can get smooth muscle contraction initiated through a variety of different ways, whereas in skeletal muscle, it was one way, acetylcholine release, right? You can read through this on your own. I probably won't. I've hit on a lot of these points, and those are the ones that I might ask you about if I discussed it in detail. And then these are just some overview slides to kind of help summarize. If I were to ask any like comparison and contrast, I think this is a good table to refer to as far as being able to look at the differences between skeletal and smooth muscle. I think this is a little more succinct and direct. All right, good luck studying chapter 7 and 8. Email me if you have any questions.